Welcome back. We're going to be talking about heredity today, specifically Mendelian inheritance and pedigrees. So, before we talk about Mendelian inheritance, let's talk about the guy that discovered it, Johann Gregor Mendel. And Mendel is considered our father of modern genetics because he created the framework for genetics inheritance that we currently use today before we had even discovered what chromosomes or genes were. He was a teacher and a monk who taught physics, botany, and natural sciences to high school and college age students in what was now the Czech Republic. So Mendel's experiment involved a couple of things. So he was growing peas in the garden, and he noticed that certain peas had certain traits, and that when he mixed his peas together or bred them together, certain traits would pass on over others. So he eventually found true breeding plants, which were ones that always passed on the same traits. And then he used those to crossbreed with other true breeding plants in order to determine what would be passed on and what trait would show up when you cross two things. And this led us to Mendelian inheritance, where we have traits that are either dominant or recessive, and they're inherited from parent to offspring. Now, in real life, not every trait follows these rules, but it's a really good foundation to start with for most traits. Now, the first thing we're going to look at is monohybrid crosses. And a monohybrid cross with a Punnett square is going to look at one trait only. And these are only valid with Mendelian inheritance for the traits that we're about to look at in the crosses. And this is going to tell us what genotypes and what phenotypes offsprings could potentially have. The thing to note with Punnett square, and I put will here, but it really should be could, is that it's a probability. So a Punnett square is going to tell you what is possible from the cross, but it can't guarantee your percentages of how many you're actually going to get because it's a 1 out of 4 in each chance. So in our monohybrid crosses, we have 3 possibilities. You can cross a homozygous with a homozygous, a heterozygous with a homozygous, or a heterozygous with a heterozygous. All right, so on the next few slides, I've mapped out kind of what these Punnett squares look like for the common crosses. And each slide is going to be arranged the same way. We're not going to go over every single one. I'm just going to go over how to read them, and then you can look at them in your own time. So on the top of the slide, it'll tell you which cross this is looking at. It'll tell you the phenotype percentages and fractions on the left, your genotype percentages or fractions, and then any notes. Note with this one that I put the dominant and the recessive. You can swap them out really easily. So I show the dominant, but if it was the recessive, then you would just flip and it would be 100% recessive phenotype and 100% recessive genotype. And then you can see those Punnett squares for the homozygous when we're crossing different types. And we've got our heterozygous in here as well. So you can again go through these on your own, and they are utilizable on our assignments. Next is a dihybrid cross. A dihybrid cross is used to look at 2 traits at a time, not just one, and it'll have 16 squares. So for these to be accurate, you must have complete dominance with a Mendelian inheritance pattern. And again, this is just going to tell us the probability of what traits an offspring could have. For our purposes, I'll never ask you about dihybrid percentages just because it doesn't always divide evenly because of the 16. So I'll always ask you these as a fraction. And typically we're going to talk about phenotype. We have 4 different phenotypes that are possible as opposed to the many, many genotypes. And then what we're looking at here is just that pattern of inheritance of, are they both going to be dominant? Are you going to have one dominant, one recessive, or are they both going to be recessive? You'll notice it's color-coded to make it easier to spot those patterns. Again, there are a couple of slides here just as examples. You will not be required to do these by hand. Any questions I ask will come from the slides so that you can reference them. And last but not least, we have pedigrees. So pedigrees are used to track traits in family trees. They can tell us the likelihood of an unknown individual having a trait, and we can use them to track a wide variety of traits from sex-linked to autosomal or dominant or recessive. So this is what a pedigree looks like. Circles are females, squares are males. If it's fully filled in, then it has the trait that you're tracking. If it's partially filled in, it's typically a carrier. Notice on this one, it's a little bit different. Your key says wild type versus homozygous or heterozygous of the other one. So this is actually a multiple allele trait. Um, but it's a similar thing when you have just a regular allele that doesn't have the multiple versions. So we have half filled in, not filled in means they don't have the trait. And then we can trace dominant or recessive traits. We can also track sex linked or autosomal. Now, you can't always tell exactly what genotype it has. This one is nice because they tell you, but if it didn't, if we were just given filled in or not filled in, then we might have to make a guess and say, well, we can look at the parents and the offspring here, and we can be sure that the recessive, we know the genotype, but if it's got a dominant, then we might have to say it could be this or that. We'll work with that a little bit more in class, but otherwise, good job.